taste the good and taste the bad, and live in healthy and live in health, So it remains in gastric cancerous tissues. So then we of course move to study the structure of these microbial communities. And then again, we have here in each line a single patient. We have here China, and we have here Mexico, and this is the level of film. You see H. pylori is a proteobacteria, so you have the highly presence of proteobacteria, and this is at the genus level, and red is Helicobacter, and then you see that, in most cases, is the most abundant in the stomach, but when you have Helicobacter decreasing in abundance, you start to see an increase in abundance of other bacteria. And so there is a great diversity in China and in Mexico. And as already pointed out to us by other speakers previously, in spite of this high diversity in the composition, if we look at the function, the profiles of function, then you see some similarity. And this was, also surprising to us to see that, between the human group, China is very, very constant, the functional profile. And in Mexico is not as constant, but it is still constant. And then it's similar between the two regions. So suggesting then that what is important then is the organ, is the physiology of the organ more important. And if you want to live in the stomach, or if you want to live in the gut, or wherever you want to live, you have to have a duty to do. And that is, to develop functions that are eligible, that are, helping to live in that niche. And so, that's what I think. But anyway, if we compare the diversity in taxa and the diversity in function with the other sides of the human body, you see we compare oral, the stool, vagina, nasal, and skin with the stomach of our patients, and we divided the stomach when H. pylori was present in red and when H. pylori was absent in blue. And then if you see these samples of the stomach are, not so like any other side of the body. Maybe just a bit close to stool, but not that much. But anyway, the diversity in the composition is more diverse. In the stomach samples, you see patients here, all over the place, not as group or as cluster as in the other sides of the body. And if you go to functions, then in functions you see more, uniformity in the different sides of the body. And again, you see a more diversity in the stomach. Maybe not as diverse as the taxa, but you still see, rather variable distribution even, of the functions. And we didn't see clearly a separation between those who got infection and those who did not got infection. Not a significant difference anyway. So what's the conclusion of this part? Well, one of the more outstanding points for us is that bacterial diversity in the stomach decreased as the severity of the lesions advanced from non-atrophic gastritis to metaplasia to gastric cancer. And our studies in microbiota indicate that H. pylori is frequent and abundant in the stomach of patients with gastric cancer. And our preliminary evidence suggests that there is some integration of H. pylori ribosomal genes in human genome. And also the comparative analysis with other body sites show higher variation in the microbiota and functional profiles in the stomach than in the other body sites. So let me move to bile duct cancer. For those who are not familiar with this, I was not familiar with this either until I got into this study. This is the common bile duct, and the tumor we were studying is in this region. So the patients we studied were endoscope, and the endoscope went from the mouth down here to the duodenum, and then when it reached the valve, this valve then by retrograde movement, then it goes up to the zone of the lesion, and then an encapsulated brush is exit, and then brush the region and get some cells and some content of the duct, and then these brush is again encapsulated, and then the endoscope is taken out. So from those samples we get we extract the DNA and then they study the microbiome. And so we studied patients with tumor in the bile duct, but we also, as a control, we studied patients who were by medical prescription, subject to also to this kind of endoscopy. And those were patients with benign disease in the biliary tract, like lithiasis or other inflammation in the site. So but not tumors. So at the level of phylum, again, you have here patients with cancer and with benign disease. And the composition was Proteobacteria, by far the most abundant phylum, almost 60% in both groups, in the cancer and in the controls. Then followed by Firmicutes and Bacteroidetes. And that was interesting to us because this is a composition which is very similar to what you see in the duodenum. So this is then suggesting to us that this what we're seeing in the bile duct might be the result of when this valve is compromised by inflammation or whatever, maybe there is a reflux of the content of the small intestine to the bile duct. That's what we think. Because at the first, what we were surprised is to find such a high amount of bacteria in the bile duct, where you have a very harsh environment, bile acid, and it's a very hard place to live, and it has consequences in the composition, as we will see later on. You want to?
Question: is this duct for delivering the bile or for reabsorbing?
For the liver, and so it's delivered to the intestine, and then it's do their thing in the intestine, and then it is reabsorbed again when it is after it is modified by the human, the gut bacteria, and then it's then absorbed and recycled. As far as I know, this is not my field either. Okay, so if we take the almost 4,000 different taxa we found in the samples, and then again do this multi-component analysis, you can see that the cancer patients group in this region and the control patients in this region, and these differences in distance was significant. And so we have a different microbial composition in cancer and the group we use as control. We did the same thing as in the other study. We asked which of the authors were the ones who gave us the most significant differences. And we got this. And I don't want to go to all of these, but I just want to point out to a few of them. Rotia is a genera that was decreased in the cancer compared with the controls. And this was interesting because this is a genus that has also been reported as decrease in skin cancer. Nestor enconia, and from Nestor enconia we got two different authors, among the most significant, in fact. And that was also surprising to us because when we were doing this study, there was no previous reports on the presence of these bacteria in humans. So it was, called our attention. And then the other one is Novosphingobin, and I will tell you why we were interested in that. Saying that Nestor enconia is in human tissues, we had to prove this. And then what we did was to design primers for a gene which is highly conserved in this species, and then with this PCR, we then went back to clinical samples and then started almost 100 benign patients and 100 biliary tract samples, and then we did PCR. And then the first thing I want you to see is that we confirmed the presence of the bacteria in a number of samples, and that by doing PCR, we also confirmed that there was a significant decrease in the presence of this bacteria in cancer compared with with the benign disease, just as we saw by microbiota results. Novel sphingomyum, it was interesting to us because we found this paper, years back, paper by Dr. Gershon in the University of California. He claimed that the infection with novel sphingomyum is associated with the development of primary biliary cirrhosis. And he says this because he found that an enzyme which is present in the bacteria, this 6-phosphate dehydrogenase, share several antigenic epitopes with the human enzyme. And this is an enzyme that is highly expressed in epithelial cells in the bile duct. So when we saw these results, we thought, well, maybe some of our patients have an autoimmune component, just as this primary biliary cirrhosis. And then we got in contact with Dr. Gershon, and then we already started serum samples from our patients and look for antibodies to these enzymes. And I just can tell you